Hello everyone, welcome back to Life of Fusion, where knowledge meets excellence. So now we are in the lecture series part 4 from Learn from PYQs for CSIR Life Sciences. So we are providing this PYQs for the practice for CSIR Net Life Sciences, and if you have any doubt while solving your questions during your practice time, you can feel free to contact us at the email ID provided in the description. So let's start the first question. The question is, the following disulfide bond containing peptide was digested using trypsin. Okay, so this is the protein connected by disulfide bond between cysteine residue, and it was digested by trypsin. So how many peptide fragments will be produced by the digestion? So you have to find how many fragments from this peptide will produce by after digestion by trypsin. So as you know, trypsin digested which amino acid? Lysine and arginine. Which side? The carboxyl side of lysine and arginine. So let's see how many lysine and arginine are here. So you see here one lysine, okay, here one arginine, then here one arginine, and here one lysine. So the breakdown will be here, okay, here and here. So how many fragments? See here, this will form one fragment. I'll let fragment number 1. Then after digestion of here, this will form the fragment number 2. This will form the fragment number 3. But see here, if this part was digested by trypsin, but still it was connected by the disulfide bond. So from this till this part will form a single fragment. So total of 4 fragments will form after digestion by trypsin. Let's come to the second question. So prostate cancer cells were treated with 2 drugs. One is drug A and other is drug B, in order to check the efficacy of the drug in arresting the growth of the cell. Okay, this was done by some researcher in the lab. They treated the cells with 2 drugs, okay, to the prostate cancer cells. So, and here they found some of the protein expression level, okay, of different protein involved in the cell death pathway of prostate cancer. So see here, drug A. In drug A, the caspase 8 level increased, beta catenin level also higher is higher, cyclin D also is slightly increased, but there is no changes observed in caspase 9. Okay, then after drug treatment, drug B. In drug B, you see there is no expression of caspase 8, not more expression of beta catenin, and there is higher expression of caspase 9, and there is also you can see there is a less expression of cyclin D. So you know that beta catenin is a transmembrane protein, okay, which was activated through Wnt pathway. Okay, so in cancer cells, okay, the Wnt pathway is to be highly activated. More activation of Wnt pathway, which leads to fibrosis or damage of the particular tissue. Okay, so most of the researcher also targets this Wnt pathway. They try to inhibit the Wnt pathway so that their drug can able to it will not progress or it will kill the cancer cells. Then you know cyclin D1 is in a cell cycle, where in the G1 phase. So this, in the presence of cyclin D1, the cells will transfer to S phase in the presence of cyclin D1. If there is no cyclin D1 or less cyclin D1, the cells will go to G0, where G0 means they will be arrested. Okay, it's called cell cycle arrest. Okay, so researchers do different strategy to target the cancer cells. So as you can see here, What drug A do? Drug A only increases caspase 8. Drug B increases caspase 9. Okay, so you know caspase 8 and caspase 9 are from two different pathway. Caspase 8 is from extrinsic pathway, whereas caspase 9 from the intrinsic pathway. Okay, when the if you see my last videos, they are also discussed some of the questions like this about Fas ligand and caspase 8 pathway. So caspase 8 from outside ligand, it will induce apoptosis. So these are the extrinsic and intrinsic pathway of apoptosis. Are you getting it? So caspase 8 include the what? BCL2 family and all. Then we can see here caspase 9, it reduce the beta catenin expression and reduce the cyclin D1, means is inhibiting the Wnt pathway and decrease the cyclin D1, so that it will cause the cell cycle arrest of cancer cells. So next come to this part. Which one of the following statement is not correct? Always when you are solving question, just see this part, which is correct or which is not correct. What they asked here, they asked which is not correct. So option 1, drug A, this one, targets Wnt signaling pathway but does not lead to death of the cell. It's not like that. It is increasing caspase 8, means it is inducing external apoptosis, but it is not downregulating Wnt pathway. So option 1 is wrong statement. 2, drug B targets the Wnt signaling pathway and leads to G1 cell arrest. Yes, drug B is targeting the Wnt signaling pathway. It was reduced, and cyclin D also reduced as compared to drug A. Means what? Cyclin D1 reduced means the cells undergo to G0 phase. G0 phase means now the cells are in cell cycle arrest. So option 2 is correct statement. 3, both drug A and drug B leads to cell death but targets different apoptotic pathway. Yes, as I told you, one is caspase 8 and one is caspase 9, extrinsic and intrinsic pathway. 3 is right. Drug A kills cell via the mitochondrial independent pathway. Yes, drug, because in intrinsic pathway is from cytochrome C, and which is from mitochondria dependent pathway, but extrinsic pathway is not mitochondria dependent pathway. So option 4 is also correct statement. So which is the correct answer here? Answer will be 1, because it asked which is not correct. Okay, let's go to next question. This is very simple question. Okay, the total number of cell in 1 ml of bacterial culture was estimated to be 2.7 into 10 to the power 6. The culture was diluted 27-fold and 100 microliter seeded per 96 well plate. What is the final cell number, they asked? So in 1 ml we have initially 2.7 into 10 to the power 6 of bacterial cells. This was diluted how much? 27-fold. So now what will be the concentration? So we have to divide it with 27. Will come 0.1 into 10 to the power 6, that equal to 1 into 10 to the power 5. Are you getting it? So now this 1 into 10 to the power 5 cells are available in 1 ml. Okay, then from this 1 ml, you took 100 microliter and was seeded in 96 well plate. So now we have to find how many cell number will be in 100 microliter. Okay, means what? 10 times less. So you have to divide it by 10. So you will get 1 into 10 to the power 4. So option 4, 1 into 10 to the power 4. Simple, don't miss like this question. Just mathematical question here. So next question. The following statement were made to suggest the existence of an enzyme substrate complex. Now enzymology. Okay. So these are different statements. A, at constant concentration of enzyme, the reaction rate increases with increasing substrate concentration. If you read the Michaelis-Menten equation, you may find that the graph, okay, your velocity, here your substrate concentration, okay. So substrate it will give a sigmoid curve. Why? Till the here means enzyme concentration is constant. At constant enzyme concentration, if you increase the substrate concentration, initially the rate of the reaction also increase. When all the enzymes were binds with the substrate, now there is no free enzyme. That time this reaction rate will be in a straight line. But here what they ask, the constant enzyme reaction rate increases with increasing substrate. Yes, but initial till this time the reaction rate will increase if you increase the substrate concentration. After some time only it will not increase, but initially it will increase. So statement A is correct. B, an enzyme catalyzed reaction has a maximal velocity. Yes, enzyme catalyzed reaction will have a highest velocity. C, at constant concentration of enzyme and substrate, an increase in the reaction rate is observed. See, when enzyme and substrate concentration is same, enzyme also same, okay, enzyme and substrate concentration is always same. So how the reaction rate will increase? It will not increase. So C is wrong statement. D, an enzyme catalyzed reaction is not influenced by high substrate concentration. No, it is influenced by high substrate concentration. If you increase the substrate concentration, okay, after some time all enzyme will bind to substrate. Now and there is no free enzyme. So at high substrate concentration, the reaction rate won't increase, it will remain in the same. So it is influence, it will influence the enzyme catalyzed reaction. So option A and B only correct statement. Which one of this above statement suggests the existence of enzyme substrate complex? 1 and B suggest the existence of enzyme substrate complex. So here they just confused you. Actually they wanted to ask you which of the following statement are correct, but they just modified that question and they asked you which of the above statement suggest the existence of an enzyme substrate complex, just to confuse you. Okay, so next question. Which one of the snRNAs given below base pair with 5' splice site of pre-mRNA? What is snRNA? You know, snRNAs, the different snRNAs are there: U1, U2, U4, U5, U6. Total 5 snRNAs are there, or it's called snRNP. Small nuclear RNA and protein, means inside the protein there is small nuclear RNA will be there. Okay, which helps in what? RNA splicing. Okay, so like U1, different RNP have, snRNP have different type of signal or different nucleotide sequence. So they binds to the specific part of the RNA during splicing process. So what is splicing? To remove the exon. So let's see, this is the exon. Okay, this is 5' side and this is 3' side. This is intron, this is intron, this is exon. This exon has to be removed. Okay, are you getting it? Sorry, this is your intron. Sorry, this is intron, this is exons. Sorry guys, this is exon. So intron has to be removed. Okay, exons will be there. So to remove the intron, we'll use what? Splicing, RNA splicing. So RNA splicing. So U1, first what happened? First U1 first come and binds to the 5' site. Then the U2 winding protein binds here in the 3' side, then U2 comes here, U2 comes and bind to U2 in the 3' side, then U4, U5 and U6, these 3 complex will come and bind here. Okay, so now tell me which one binds to the 5' side? U1 binds to the 5' side. So hope you like this video, and we'll upload so many more video on learn from PYQs. So you can subscribe our channel, and we have also a playlist for learn from PYQs. You can follow that so that you'll get all the updates on different PYQ questions which will solve and upload. Thank you everyone. Have a good day.